Hello, welcome to Pharmacognosy and Metabolic Engineering. This is NPTEL online certification course. So, now come to lecture 23, where I will talk about indole alkaloids, the environmental factors that regulate indole alkaloid biosynthesis. And under this, what I am going to cover basically the role of light and elicitor in upliftment of indole alkaloids in culture, and cherry root culture system for indole alkaloid production and to study the metabolic pathway, and finally, we will also see the elicitor induced expression of early pathway genes in indole alkaloids biosynthesis. That means, how elicitors can upregulate the indole alkaloid production. So, I would like to tell that so far our discussion we are mostly dealing with some of the only early steps of indole alkaloid biosynthesis, but the pathway is very complex and in the subsequent classes we will look into it, but it is interesting also. So, do not get daunted. So, now go to the let us go to the board. So, board is this, that the plant cell and tissue culture system for indole alkaloid production. I have already mentioned that cell cultures of Catharanthus roseus was explored, but it failed to produce the in late enzymes of the pathway. So, let me briefly remind you the final stages of the biosynthesis. That means, what I said, that ultimate target is basically to find vinblastine or vincristine from catharanthine and vindolin, and that eventually coming from streptocidin, and which is basically coming from secologanin and tryptamine. And in between also there are products like azmalicin and other product, canthenamine also on. So, cell culture failed to produce the alkaloids like vindolin and catharanthine, which are monomeric indole alkaloid, and I have made it clear in the earlier class that vinblastine and vincristine, these are dimeric. So, then cell culture failed to produce vindolin or even catharanthine. So, from there scientists thought that maybe differentiated cultures plays important role. So, they go for differentiated culture. One good example of differentiated culture is basically shoot culture. So, and another one will be the hairy root culture. With hairy root culture, there are reports which are not always consistent with the findings from different groups. For example, one group in United Kingdom, they have developed hairy root culture of Catharanthus roseus, and using immunoassay based detection, they could detect that vinblastine in the hairy root culture. Detection of vinblastine, UK group, whereas the group from Netherlands or in USA, they did not find any vinblastine. Okay. But then what they have found in hairy root cultures? Hairy root cultures are capable of producing some of the early indole alkaloids, and also hairy root cultures are also capable of producing a few late indole alkaloids of the pathway, which is called tabarsonin. So, where tabarsonin will come? Tabarsonin will come here. So, I will write tabarsonin now with the pen. So, late indole alkaloids, for example, tabarsonin detected, but this is detected in hairy root cultures when they are grown under dark condition. Okay. But no vindolin was found, okay, and some catharanthine are also detected. Now, let us see what is happening with the multiple shoot cultures. So, the multiple shoot cultures appears to be a very promising system, and here scientists they have detected vindolin accumulation in substantial amount, and not only that, one of the multiple shoot cultures also reported of vinblastine accumulation in this multiple shoot culture. So, now where lies the difference? So, multiple shoot cultures, they are basically when we are talking about shoots cannot grow in under dark condition. Shoots cannot grow under dark conditions. So, in order to grow shoots, you have to put light. That means, the question comes here, the light, whether light plays important role in this vindolin accumulation or not. Okay. So, when that is the case, one interesting experiments were conducted by the group from Rice University, US. So, what they have done, they already developed very stable hairy root culture. These hairy root cultures, they made them photoheterotrophic. That means, they slowly reduce the percentage of sugar, and instead they are giving them sufficient light illumination. And under that condition, what will happen? These hairy root cultures will turn green. So, their idea is basically the whether green hairy root culture are capable of producing vindolin or not. If they are producing vindolin, then that is good. So, that may be a serve as a good source for vindolin, and then catharanthine can already produced in hairy root culture by elicitation. So, if the green hairy root cultures produce vindolin, then vindolin can be harvested. But to their surprise, green hairy root culture are not producing any vindolin. Instead, they are producing the other alkaloids. So, what happens that the green hairy root when the normal hairy root cultures are grown in dark, what I said, they produces tabarsonin, and then they checked what happens to the tabarsonin condition. In the green hairy root culture, what they have also found that no tabarsonin. That means, in green hairy root culture, tabarsonin formation is not there. If tabarsonin formation is not there, vindolin cannot be formed. So, from there what is clear to us, that tabarsonin requires a dark condition. Tabarsonin synthesis cannot synthesize. So, under light tabarsonin may not synthesize. So, that is why when hairy root cultures are grown under dark, it accumulates tabarsonin. But green hairy root cultures cannot produce vindolin. The reason is that it is not producing tabarsonin. So, how can vindolin will come? Okay. So, this is an important point. So, next what we should see, that what happens when plants are getting light. But that basically that we will cover, but I am not covering that aspect now, because when I bring these issues here, then that leads to the dissection of the late steps of vindolin formation from tabarsonin to vindolin and beyond. So, that I will look in one of the subsequent classes. So, what I am going to see now, that let us go to the concept of cell culture. So, the cell culture failed to produce vindolin, catharanthine, but they are producing azmalicin, azmalin, serpentine, all these things. So, these alkaloids are also important. Catharanthus roseus producing so on. So, next what they did, the scientists, they try to produce more of this in cell culture. So, how they can do that? So, in one of the earlier classes I mentioned about elicitation. So, that means we will bring that issue again here in this class, that is elicitation of cell and organ culture. So, this organ culture also includes hairy root culture for enhancement of alkaloids. So, here I will before I go to the elicitation mechanism, I will give you some good example of the practical work. So, that means this one US group. So, what they have done? So, as I said that they have a very stable hairy root line. So, hairy root culture of Catharanthus roseus was made and was treated with different elicitors. What are these elicitors? These elicitors are they have chosen, one is pectinase, another one is chitin, and third one is jasmonic acid. So, I will write here elicitor treatment. Now, here something I have to tell, that this elicitor treatment how it to be done. So, once you have the cell culture or organ cultures growing and it reach the late, this is L, least exponential phase. So, at that stage elicitors can be applied and the duration may be for 48 hours, and then cultures to be harvested and look for the or detection of, oh my hand is sleeping. Mostly the HPLC based methods are using. So, they use TLC, HPLC, DAD, and sometimes they use mass spectrometry to confirm the products. So, this is how it has to be done. So, pectinase is basically an endogalactoduinase. So, upon application of pectinase, what happened, that it increased the level of tabarsonin. So, tabarsonin level increased by 2.5 times than control culture. Okay. Upon application of chitin, chitin is basically a component of fungal cell wall. So, when you apply chitin, so the system will think that they are under fungal attack. So, they will induce the specialized metabolism, and as a result you will see the enhancement of alkaloids. So, here basically this lead to increase of azmalicin level up to 50% when you compare with the control. When jasmonic acid was used, so jasmonic acid is basically a signal transducer in defense response. So, it basically increased specific yield of azumalisine by 80% and serpentine by 60%. Not only that, they have also found two alkaloids which are normally the late step alkaloids, which is called lochnadicin, LOCH NARICINE and HOR So, I just check the lochnadicin, lochnadicin and horhamycin. So, these are basically the late step of indole alkaloids which are produced only in roots. So, they also found that there is an enhancement of these alkaloids as well when treated with jasmonic acid. So, jasmonic acid was found to be the most suitable elicitor for upliftment of alkaloids, and this is interesting. Once you treat with the elicitors, the pathway uplifted. So, the enzyme activity can be studied and subsequently genes can be isolated. So, in the next board I will show you what happens upon this elicitation. So, apart from this jasmonic acid, even the and fungal extract, even yeast elicitors can also, yeast extracts can also used as elicitors. So, we also study the biosynthetic pathway, and we have also used the yeast elicitors for our own experimental work, particularly for upliftment of phenolic compounds. So, so that means that yeast extracts, filter sterilized yeast extracts or autoclaved yeast extracts can be used as elicitor. So, what happens? So, in this elicitation process. So, that I would like to tell, but before I will tell that what happens upon this in this signal transduction process upon jasmonate treatment. So, there are two things. So, let me draw this model for elicitor leading to STR gene expression. Okay. This is the plasma membrane. Here lies the receptor so where elicitor molecule it come and sit over here. And that leads to winning of ion channels and influx of calcium. This is cytosol, this is inside the nucleus. So, here this is JERE, Jasmonate elicitor response element, then there is a TATA, and then the gene for STR is here. So, if it works, that means for from lipase through this octadecanoid pathway jasmonate are synthesized So, here lies the ORCA. So, and so what happens that when any means apart from jasmonate any other elicitor is used, okay, say it could be fungal extract, say it could be yeast extract. So, that binds and that opens the calcium channel, and that calcium channels directly influence the BA area, that is where the elicitor response elements, where this 1 transcription factor binds, which is called CRBPF1. So, the CRBPF1, CR stands for Catharanthus roseus, BPF1, this is Catharanthus roseus P box binding factor 1 homolog. Catharanthus roseus P box binding factor 1 homolog. So, this is 1, and the ORCA, this is octadecanoid responsive Catharanthus AP2 domain protein. So, this again I will mention in the next class as well. So, what happens, there are two ways, one is via calcium channel, it activates one transcription factors which bind in the elicited response element, and other one by the activation of the jasmonate pathway, which activates the jasmonate receptor, and then protein kinase plays role, and then what happens, that activates a specific transcription factor which is called ORCA, octadecanoid responsive Catharanthus AP2 protein. I will mention that later, and that binds to the particular jasmonate elicited response element here, and then as a result of that it initiates the transcription, and we see that STR genes are being transcribed. So, normally what happens, sometimes the specialized metabolite levels are very low in cell and organ culture, and that level can be enhanced by adding the elicitation. So, elicitation is basically activating the pathway. So, this is the early step, and as a result of that there will be more transcription leading to more enzyme protein, and depends on the substrate availability, it ultimately leads to more product formation. Product formation in higher amount. So, I will end this class here, and the next class we look into more details at the molecular level, how these specialized transcription factors bind and modulate the process, and once we finish that, then we will move into the late steps of indole alkaloid biosynthesis, that will be the subsequent classes. So, with this I stop here. Thank you very much.